Have you ever wondered how drugs are made? Do you know how we know the quantity that humans should take to achieve the desired result? There are so many diseases in the world. Cancer is an example. It's actually a major cause of death globally. And now there are so many anti-cancer drugs. You have the cisplatin, the doxorubicin, and what have you. And for each type of cancer, there are different dosages. For instance, ovarian cancer, you're told to take 100 milligram of cisplatin. For testicular cancer, you're told to take 20 milligram of the same cisplatin. For bladder cancer, you're told to take 50 milligram. Now, do you know the process that researchers go through to actually arrive at that conclusion? Or do they just wake up and say, oh, take this amount?
Now, in the drug development process, there
are different stages. There are 5 stages. The first is identifying the potential target. Maybe you are a researcher and you've discovered that this small molecule or this peptide, has some property, has some antimicrobial property, or has some anti-cancer properties. Now, do you know how to know the amount that humans should take to achieve the desired results? So the first stage, drug identification, identifying the potential target. Now, the second stage is preclinical trial. This is where you take the drug, go to the lab, and treat the cells or the microorganism or the bacteria or whatever biological system it is. So what you're trying to find out is the IC50. IC50 is the concentration that is needed to kill or to inhibit 50% of that biological system. In the case of cancer, you take cancer cells, you go to the lab, you treat it with the drug that you've discovered, maybe cisplatin or maybe a new drug that you're working on. You treat it and you find the IC50. When you have found the IC50, you can go ahead to treat animals, okay, and see if it's actually working. If you get a positive result, a good result, now you can go ahead to test a small population of humans. Now, this is the third stage, that's the clinical trial. Test a small population of human beings. Now, if you get a good result that, okay, it's actually working, it's actually killing the cancer cell, you can go ahead to seek for FDA approval. If you get it, that's good.
And the fifth one is the drug manufacturing, after which they are released to the populace.
Doctors are going to start prescribing it and all of that. Now, after this stage, you want to also get feedback to see if it's actually working, okay, or not. Now, in this video, I'm going to be showing you how we determine the IC50 of anti-cancer drugs. My name is Olebube and I'm a third-year PhD researcher at King's College London, Department of Chemistry on Schneider Group. So today I'll be showing you what cancer cells look like and the process we go through to determine IC50 of drug. Okay, so come with me.
Ensure to wear your PPE before entering the tissue culture room. Thaw the cells by gently swirling in the water bath. Add medium to the cells and transfer to a centrifuge tube. Centrifuge for about 5 minutes to pellet the cells. You can see the cell pellets at the bottom of the tube. Carefully remove the supernatant and resuspend the pellets in fresh growth medium. Transfer the resuspended cells in a flask and place in an incubator. Monitor the cells to ensure they are growing properly. When the cells have reached about 80% confluency, you can check this by viewing under the microscope. Go ahead to count the cells and then seed in a 96 well plate. Your seeding density should be determined by the type of cell you're working with and your experimental design. Incubate the cells overnight to allow for attachment. Check that the cells are attached before treating with the drug the next day. When you are ready to treat the cells with your target drug, prepare a stock solution of this drug by dissolving in a calculated volume of the appropriate solvent. Use a nanodrop to confirm the concentration you've made and then dilute to your maximum dose using the growth medium. Serially dilute your maximum dose to have a range of different concentrations. I will start by labeling another sterile 96 well plate according to the dilution series I want to use for treatment. I would add 100 microliters of the growth medium to the wells in column 1 to 11 and add 200 microliters of the growth medium to the wells in column 12. I will mix my highest concentration and transfer 100 microliters to the nearby wells containing only medium. I will mix this first solution and transfer 100 microliter to the nearby wells containing only medium. I will repeat this process until the final concentrations will be a series of dilutions. I will then retrieve the attached cells from the incubator, remove the old growth medium by pipetting, and transfer 100 microliter of the different drug concentrations to the wells containing the cells. The first column will be my control wells without any treatment and will indicate the maximum cell viability. I would then transfer the treated cells to an incubator for 24 to 72 hours depending on my experimental design. I would add 10 microliters of the yellow MTT reagent to the treated and untreated cells and incubate for 3 to 4 hours. I would then add 100 microliters of the MTT solubilization buffer to dissolve any formazan crystals. The reduction of the yellow MTT reagents to purple formazan crystals occurs within the mitochondria of live cells. Cells with little or no treatment are usually purple in color, while those treated with high doses of the peptide are usually yellow because there are no metabolically active cells to reduce the MTT reagents. The number of viable cells is directly proportional to the intensity of the purple color and is measured spectrophotometrically at a wavelength of 570 nanometers. The absorbance values are plotted against the concentrations to get a dose response curve. The IC50 is the concentration that results in a 50% reduction in cell viability compared to the control.
I hope you found this video useful. If you did, let me know in the comment section, and also let me know what you want to see in my next video. Please like, share to every researcher you know, and I'll see you next time. Bye!